Hey everyone, Sydney Sozio here. Thanks for joining us on another episode of the Quarantine Classroom. This week's topic is DNA mutations, more specifically Frameshift mutations, and then learning how to decode amino acid sequences. Let's start with some basics. Proteins are made up of amino acids that are composed of base pairs of DNA. These base pairs are pairs of nucleotides like adenine and thymine or cysteine and guanine. Mutations occur at random, and there are a few different ones, but today we're going to talk about the Frameshift mutation. This happens when a single base pair is added or deleted from the sequence. Another important thing to note is that codons are read in sets of 3, so if one letter is added or deleted, the window that is read is going to change for every single nucleotide down the line, which then impacts the entire rest of the chain and all of the proteins that are made. Depending on where in the chain they occur, this one mutation can cause the majority of a chain to code for the wrong proteins and could render the entire chain useless. And now I'll show you exactly how I got this. It should be easy to follow, but I have the attention span of a goldfish, and also I did all of the work for this exclusively between 11 p.m. and 3 a.m., so I hope it makes sense. First, this is everything I was given. I went into Google Docs and formatted this document to organize data because I am very much a visual person. I made 2 tables, one for the normal strand and one for the mutant strand of proteins. I started by labeling the top row with the proteins broken up by codon for each strand. Both strands start with codons AUG, which is a start codon, and then UGG. The third codon is where I noticed the difference between the 2 strands: phenylalanine and isoleucine. I looked at the original strand, and both options start with UU. Isoleucine's options all start with A followed by UU, and since I know it's a frameshift mutation, I can assume that A, or adenine, is the one that's added here, so the rest of the base pairs for the mutant strand have to follow the same order as the original strand. Unless there is another mutation, but for this specific problem, I knew there wasn't going to be. Since I know phenylalanine can be either UUU or UUC, I looked at the options for codon 4 in the mutant strand. Both started with a C, so I knew the sequence for phenylalanine had to be UUC. I went back up to the normal strand and saw all of codon 4's options started with AC, so I knew codon 4 in the mutant strand had to be CAC. Another thing I was tipped off to in the directions was that there was a stop codon in the mutant strand. The normal strand also kept going with an ellipses and the mutant strand stopped, so I figured the stop codon was after codon 4. I looked back down at the mutant codon 5 and saw all of the options started with U, so codon 4 had to be ACU. Codon 5's options in the normal strand both started with AA, and there's a stop codon UAA that would correspond to that. So once I figured that out, I didn't really need to figure out the rest of the strand because the question only asked for the mutant mRNA strand. Thanks for sticking around to learn how to deduce a coding sequence. That's all I have for you this time, but I'll see you next week.